Welcome to video 1 of a two-part video series on genetic material and the cell cycle. Please get out your big sheet and write on your big sheet as I'm writing on my big sheet. Feel free to pause when you need to take time to catch up. We'll be starting on the genetic material side of the big sheet that has to do with DNA and chromosomes. I want to start with this diagram first because it gives a really good overview of DNA and chromosomes and how those are related to each other. This X-shaped structure right here is a duplicated chromosome, and it's made up of tightly coiled DNA. It's a replicated chromosome because it has two sides. As you begin to unravel this chromosome, you find slightly less coiled DNA called chromatin. Chromatin is uncoiled DNA organized around histone proteins. The histone proteins act kind of like a spool for thread, and then the DNA ravels around the thread to keep them from getting tangled. As you continue to unravel the DNA, you see this structure of DNA, which you all know is called the double helix. If we continue to zoom in on the DNA, we see the DNA nucleotide sequence. This DNA nucleotide sequence codes for proteins. So the sequence or order of nucleotides code for proteins. And every 3 letters equals an amino acid, right? Those are called codons. Now, these nucleotides, can be different, but can code for the same trait. That's called an allele, An allele is a different form of the same gene. So this nucleotide sequence right here could code for height in pea plants. And one set of code could code for a tall pea plant. Another set of code could code for a short pea plant, but both of them code for pea plant height. When we say types of genetic material, we're basically saying different ways to package DNA, because our genetic material is DNA, in living organisms, and then we use RNA as well to take information out of the DNA, but our primary genetic information is DNA. When we look at this cell right here, this is the nucleus of the cell. That's how we know that it's a eukaryotic cell. And inside the nucleus, we find DNA in the form it spends most of its time in. This uncoiled DNA in here is our chromatin. This is the form we can use most of the time, and this is the form of DNA we unravel to read to make proteins. So most of the time when a cell is doing its job, it's in the form of chromatin. Chromatin is found in G0 when the cell is not in the cell cycle. This is when the cell is doing its job, going about its normal life. So most of the time, when a cell is doing its normal job, the DNA is in the nucleus in a form called chromatin. Chromatin is also found during interphase, because then it's open enough to be able to copy itself. DNA can also be found in duplicated or replicated chromosomes. So we can call these coiled replicated, often known as duplicated chromosomes. A duplicated chromosome has 2 sides. Here's one chromatid. Here's another chromatid. So a duplicated chromosome is made up of 2 chromatids. And the middle narrow part is called the centromere. Now, each of these chromatids is made up of a long strand of DNA. So this chromatid is a long strand of DNA, and this chromatid is a long strand of DNA. And the sides are identical to each other. Okay? So for example, let's say that the top side of this chromosome had a large B gene. That means the top side of this chromosome would have a large B gene. Let's say the bottom side of this chromosome had a large T gene. The bottom side of this chromatid would have a large T gene as well, right? So the chromatids are identical to each other. So this right here is one chromosome. You find chromosomes in this form during late prophase of mitosis, during like metaphase. So basically, like when the chromosomes are starting to like line up in the center of the cell, that's when you find duplicated chromosomes like this. Now, let's say that this chromosome and this chromosome are something that we would call homologous chromosomes. Okay. So let's say these two chromosomes are homologous chromosomes. What is a homologous chromosome? A homologous chromosome is a chromosome that has the same type of information but different alleles. So for instance, while this chromosome right here in red carried the big B and the big T gene, we could have these chromatids carry the same type of gene but not identical genes. So for example, this one could be little b, and this could be little t, little t. So they're carrying the same types of genes, but they're not identical. So homologous chromosomes, and I'm actually going to write what a homologous chromosome is down here because I ran out of room up top. So a homologous chromosome is a chromosome that has the same type of gene but different alleles. Okay, so this green chromosome is homologous to this red chromosome. Okay, the two sides of this red chromosome are part of the same chromosome. They're identical to each other. Now, the last thing I want to look at on this side is these coiled unreplicated chromosomes. Much like the duplicated chromosomes, they have genes on them and they're made up of long strands of DNA. These coiled unreplicated chromosomes can also be called coiled unduplicated chromosomes. Okay, because they only have one side. This is what you have before the cell cycle begins. So for example, if this was the red chromosome, it would have the big B gene and the big T gene. It just wouldn't have another side to it. It's unduplicated. It hasn't copied yet. You have to duplicate the chromosomes so that the two sides can be split up during mitosis. Here's another unduplicated chromosome, right? So here's the little b and the little t. That means that these two chromosomes, right, have the same type of information but not identical information. So these two chromosomes right here are homologous chromosomes. You see unduplicated chromosomes before the cell cycle begins. Before the cell cycle begins, kind of. You really, well, not really. The DNA is unduplicated then, but you wouldn't actually see the chromosomes. It'd still be in chromatin form. You really see unduplicated chromosomes during anaphase and during telophase. Okay. So why is this information important? This information is important because your chromosomes have all your genes on them, and your genes contain genetic information that make your physical appearance. They help make neural connections, so they kind of make your phenotype, the physical expression of your DNA. And like I said before, that's coded for in alleles. Okay, we all have different alleles. And alleles, remember, are different forms of the same gene. For instance, here's an example. Here's chromosome 1, okay, and it has allele for purple flowers. Here's another chromosome 1, and it has an allele for white flower color. So these two chromosomes both have an allele for flower color, but it is a different color. That also makes these two chromosomes homologous to each other. Homologous chromosomes can be duplicated or unduplicated. Sometimes doctors are concerned about genetic disorders, so they might pull something called a karyotype. A karyotype is a chart of chromosome pairs. And in a karyotype, they're looking at the chromosomes to look at the banding pattern to make sure that you have the right number of chromosomes, to make sure that your chromosomes aren't missing pieces. This chart shows unduplicated chromosomes here. I can tell because each chromosome only has one side. So you see in chromosome number 1, here's one copy of chromosome number 1 you got from one parent. Okay. Here is the other copy of chromosome number 1 you got from the other parent. So these two chromosomes are homologous to each other. Same thing for chromosome pair 2. You got one copy from one parent, you got one copy from the other parent, and so on. Same thing with chromosome 3, and so on. So humans have 46 chromosomes, okay, 23 pairs of chromosomes. Chromosome pairs 1 through 22 these are known as autosomal chromosomes. And chromosome pair 23 right here, these are known as your sex chromosomes because they determine if you're born with female or male secondary or sex characteristics. Okay? So this child is XY, and so that child would be considered male. So scientists can look at a karyotype and see if there's anything wrong. For instance, or not typical. Okay, for instance, if you have an extra chromosome 21, okay, that would be an indication of Down syndrome. This concludes part 1 of the video series. Please make sure you've written down all the information on your big sheet and come with questions in class. We will stop this video and please watch the next video, part 2, to review mitosis. Thank you so much.